Hey everybody, it's Miss Hunt. Today is Tuesday, December the 1st, and I hope you all are doing well and staying warm. All right, so let's go through this week's Module 13 and talk about all the stuff we've got to do. Our lesson plan right here. Here's our lesson plan for this week. Yesterday, we did meiosis drawing, and we didn't quite finish it in one of my classes, but the other one we did. And we watched the Amoeba Sisters video, and we did a little worksheet that went with it. There is a crash course that I want you to watch the video, and if you do the guide with it, you get 2 points extra credit. Today we have, we're going to finish the drawing in one of the classes, but then otherwise we have some notes on meiosis, and I'm going to go through that with you. I'd like for you to watch the video on the AP Classroom tonight for homework. Tomorrow you have a POGL to do on meiosis, and there's another video on 5.2 to watch. We have a genetic diversity lab to do on Thursday, and you're going to study for your quiz on Friday, which is in the AP Classroom, which we'll be taking in class because we'll be here on Friday. Of course, if you're online or absent, then you'll be taking it at home. All right, and just a reminder for any online kids, online learning ends this semester, so it does not go on to second semester. All right, and then we have 2 more weeks and we are done for Christmas break. So we're going to try to push forward and get everything done that we need to get done. So I'm kind of excited about that. Okay, so yesterday we did the Amoeba Sisters, and I need to put a video link in here. I haven't done that yet, but I will. Watch the video and do the worksheet. It's very easy to do that. And let me know if you have any questions with it. It should be pretty self-explanatory. All right, the drawing. So here's the doc right here. If you're at home, it could be a difficult thing to draw this online. Okay. If there's a way for you to print it out, that would be the best, to print this out and then practice drawing through it. It's not for grade, but it's for learning. So it's really important for you to learn how to recognize all these phases. So here is the finished copy of mine, and it's what I used to teach from. Of course, in class, I did it on the document camera, and the kids kept up with me as they could do it with me, but we were looking off of this right here. So let me go through it with you, and hopefully you have a chance to draw it out. You could draw it out on paper so you can recognize what's all happening. I think when you draw it and you look at it, you start to learn it a lot better. Okay, so we're going to start up here. I did it separately up here at the top, so you can understand. Normally you have one of these blue and one of these pink from your mom. And let me look real quick. What those are chromosomes. So I want you to see, this is a normal, no, not that one, I'm sorry. Sometimes they don't do them right. Okay. So this is a normal human karyotype for a human chromosome, what they're supposed to look like. Okay, let me move me real quick. So I just Googled this right here. It's a normal human karyotype. So you get one of these are the chromosomes right here. And if you counted, you would have 23 pairs. So that's 46 chromosomes. So you have chromosome pair number 1, and you notice that they're the same length, and they have the same genes, the same markers, like that's a white right there, there's a gray, and there's a black. They have the same genes on there. So what this is a very tightly wound up piece of DNA. On the DNA, of course, it codes for a protein, and that protein codes for you. So if it codes for your hair, your color of your skin, or the shape of your jaw, whatever it is, your heart, it's all in here. And you got one chromosome from your mom and one chromosome from your dad. And you can see all the way down here, they all match up. And then this last pair is your gender, your 23rd chromosome. So this right here, we're looking at a boy. One long chromosome and one short chromosome makes a boy. If it was 2 long chromosomes, it would make a girl. Okay. All right. So when we go back to my picture here, what we're looking at is that would be one from dad and one from mom. And we just colored them colors for gender. So in the S phase of interphase, the chromosomes double. So now I have 2 identical matching ones from dad and then 2 from my mom. That's why it looks like an X, but really they're like 2 V's next to each other. And they're tied together with the centromere, which is in the middle. So I said, let's pretend like the gene for hair is right here. So it's the identical one right here. So dad's color of hair is here. And then that would be mom's gene for hair and whatever color she has, and it's doubled here. This right here, 2 of my dad's, so that doubled chromosome, that is called a sister chromatid. And together, these from dad and mom, they make the homologous chromosomes. So remember, HOMO, homo means same. So they have the same traits in here, the same genes on here. We really say that there's an allele from dad and an allele from mom, and that's what you're going to be inheriting. So it's important to recognize sister chromatids and homologous chromosomes. That's going to help us throughout this whole thing. All right, I drew our, what we call the original cell right here. So our original cell, we're going to use 4 chromosomes because I don't want to draw 46 chromosomes. That would take forever. So we're going to say that these are homologous. So I have 1 from my mom and 1 from my dad. So we could say this is like chromosome pair number 1. So what I'm saying is like this right here, 1 from my mom and 1 from my dad, chromosome pair number 1 of those 46 chromosomes. And then over here, this could be chromosome number 2 or another short one if you wanted to put it like as a 13. They get shorter as they go through here. But I just chose 2 little ones. Okay, in the S phase of interphase, the DNA gets synthesized and doubles. So here my chromosomes have doubled. Okay, now this is mitosis. So mitosis is what our somatic cells are. Our body cells do this. We just finished mitosis from the week before Thanksgiving. So it's kind of a review for mitosis. We have our centrioles at the poles. The nucleus starts to disappear. The sister chromatids come together. The sister chromatids line up in metaphase. Anaphase pulls apart the sister chromatids. Telophase, the nucleus starts to come back and it forms. And we have 2 daughter cells that are identical to the parent. And those are your somatic body cells, my heart, my liver, my blood, my kidneys, every cell in my body, except for my reproductive cells. All right, so you should understand that. Now, this week we're doing meiosis. And let me say it correctly for you. The word is meiosis. Meiosis. In the South, we have a hard time saying our T's and D's. So mitosis sounds a little bit funny. And when the teacher says mitosis and meiosis, they sound a lot alike. So I often will emphasize the E in meiosis and say meiosis. And I encourage you to put the word egg, like EGG, and the S put sperm, SPERM, so that you're reminded that this is a reproductive one. So this is for making our gametes, which are egg and sperm. And what cells do this? The ovaries on a female are going to make eggs, and the testicles on a boy will make the sperm. And it's going to have half the number of chromosomes. So I'm going to take my original cell here. So if it's a female, this would be an ovarian cell. And if it was a male, it'd be a testicular cell. All right. And in the S phase of interphase, because everybody still goes through interphase, in the S phase, we're going to have doubling happening. And then in prophase, we have a magical thing called crossing over that happens. So let's remind ourselves, these are homologous, meaning that they have the same information on them. They've just doubled. So if I look at my normal one, I'm seeing 2 of these and 2 of these. And what happens with crossing over, a piece of the one from your one parent switches with the other parent. And that gives us that crossing over. I didn't cross over on my little ones just because I didn't want to have to keep drawing it over and over. So we switch just the big ones. Now, what part switches? It's random. How much switches? It's random. Could it be a whole piece, a little tiny segment? It's random. There's no reason why, and they don't always switch over. But what this means is if my child inherits this chromosome, he will have most of what my dad has, my traits from my father. If this chromosome is the one that gets inherited, the child will inherit a little bit of my dad's DNA, but also a piece of my mother's DNA. So why does this happen? Well, crossing over happens to guarantee variation in a species so that we don't look identical to our parents and we have a better chance to mix up the DNA. Everything in genetics shows me that you're supposed to be a unique individual. You're not supposed to look like anybody else. You're supposed to be unique, which is kind of a nice feeling. Okay, crossing over only happens in prophase 1. That's it. All right, metaphase 1, look here and see how these are the homologous chromosomes that are lining up in the middle here. This does not look like it does up here where we have the sister chromatids stacked on top of each other. Okay, here we have the homologous chromosomes lining up. In anaphase, we're pulling those homologous chromosomes to each pole. We're not pulling the sister chromatids apart. So notice that that looks very different. In telophase 1, my nucleus starts to reappear. Remember, these are sister chromatids inside here. Okay. Really, there is a brief cytokinesis, and then we go right into prophase 2. In prophase 2, the nucleus falls apart. The sister chromatids are still in pairs. We go to metaphase 2, and notice here I have 2 cells. First of all, you'll see all of meiosis 2 has 2 cells, but you see that metaphase, the sister chromatids are lined up. So metaphase 2 looks the most like mitosis compared to mitosis. So you see the sister chromatids are lined up. The spindle fibers are pulling them apart. Anaphase, the sister chromatids have separated. So if it says sister chromatids, we're in metaphase 2, or we're in meiosis 2. If we're talking about homologous chromosomes, we're in meiosis 1. Anaphase away, sister chromatids get pulled apart. We go to telophase where the nucleus starts to come back. And then we are left with 4 cells, and they have half the number of chromosomes. So my original has 1, 2, 3, 4. Down here, I just have 1, 2, right? Remember that these are called haploid, meaning half the number of chromosomes. These up here are diploid, having the full set of chromosomes. We call this N, which means it's the number of chromosomes. And up here, we call these 2N, meaning they have the double the number of chromosomes. These are not identical to each other, and they're not identical to the parent. These are gametes. So if this is a female, then each one of these is an egg. And if it's a sperm, then each one of these is one sperm. And they would be housed in the testicle or housed in the ovary. And when this individual reproduces, if it's a female, 1 egg will come out at a time, and it can be fertilized by the sperm. There's a lot of sperm coming out at one time, but each individual sperm cell is unique and different than the other ones, which is pretty interesting. So which egg is it that is available to be fertilized is random, and then which sperm meets it is random. So it, again, tries to give us variation. So let's just look at this. If this is the egg that gets fertilized, then the offspring would inherit their grandmother's gene with a little bit of their grandfather's and all of their grandfather's gene here. If this is the egg that fertilizes, the child would inherit all of their grandmother's gene and all of their grandfather's for the second trait. So you can still see they have enough of the information for hair, if that's what we wanted to say was on the big chromosome was hair. They would still have the trait for hair color. It's just they would inherit just the grandmothers or grandfathers or grandfathers. So that's pretty interesting to think about which ones you inherited, which ones your parents passed on to you. All right, so after we finish that from Monday, Amoeba Sisters and this, okay, today we're doing 5.1 meiosis notes. So I'm going to go ahead and run through that really quick so that I can go through that with you. Close. Okay. Okay. All right, so remember meiosis, we're going from diploid, which is the full set of chromosomes, to haploid, which is half the number of chromosomes. All right, that's just our standard for you to go back. And remember, with meiosis, we're talking about sexual reproduction. So not everything does sexual reproduction. So we have to understand that if it's not sexual, it's not meiosis. It just does mitosis. So our bacteria are just going to do asexual reproduction. So why is sex important? So remember, we're talking about variations in the species. Asexual, you get a quick reproduction. You do not use a lot of energy to find a mate or anything like that. You don't have to generate the gametes. Most of them are not, or all of them are not going to take care of their offspring. It's very quick. So we're talking about fungus and bacteria. Some plants do asexual reproduction. Sexual reproduction is very common. It's expensive because you have to build the gametes, you have to house the offspring, you have to make those egg and sperm, you have to find a mate. A lot of times there's a lot of battling between mates in the mating season that can happen. So this is kind of a question of then why, if sexual reproduction is so problematic, why not just do asexual reproduction? All right, well, what are the advantages of having sex? There's a good video here, and I'd like for you to watch it. It's an hour long, and it talks all about the variations in those species. So it's very good, but I'm not showing it right now because it's an hour long. But please watch it on your own. All right, asexual reproduction, single individual is the sole parent. They're going to pass down all their genes, so they're identical. This is great for like strep throat. Strep throat is caused by streptococcal bacteria. So the bacteria reproduces asexually. So if I get strep throat and somebody in India gets strep throat, it's the exact same looking bacteria. It's around the world. It's easy to test. It's easy to make a test for it. And it's also easy to cure it. We know what antibiotics work to kill strep throat. So it's a very easy to destroy bacteria. So even though it grows really rapidly, it's easy to identify. They're all clones to each other. Sexual reproduction, you have two parents with half the genes. We get a unique combination. So like I said with my strep throat, one antibiotic can kill all of it. But if we have sexual reproduction, our offsprings are different. So one antibiotic wouldn't kill. So think about it like as a pesticide. Of course, we want to think of pesticides in both ways. One way is we don't want the pests. They're eating our crops or something like that. And the other way is it's not good to kill all the species. So if I put a broadband spectrum pesticide out and it kills almost all the insects, but maybe it doesn't kill all of the crickets. Those crickets have a genetic diversity, and those crickets allow them to survive that pesticide and reproduce. They are pesticide resistant, which may or may not be good, but because they had a variation, they're able to survive. Right now, we're talking about COVID, and we see variations in the species right now. Some people get COVID and they get really, really sick and they have to go to a hospital. And other people get COVID and nothing happens to them. So that is by your genetics, whether you are going to get sick or not. It's variations in our species, whether you can bind that virus or that virus doesn't even come in and bind to you. So it's just a variation in our species, which is good for survival. All right, fertilization and meiosis, we alternate the sexual life cycle. So wherein adults come together to make sperm and egg, we have fertilization, and then we make that zygote, which is the one cell that is diploid that has a full set of chromosomes. And then that goes into being a child, which is still diploid. So what it's saying is we spend diploid and then haploid in our life cycle. The gametes meet. That's what I say. This is egg and sperm. So I think about the E and the S for egg and sperm. Gametes meet. Similar chromosomes that determine the individual's sex. Females have homologous chromosomes of a pair of X chromosomes. Males have an XY. And thus humans have 22 pairs of autosome and 1 pair of sex chromosomes. So autosomes are just all the chromosomes. The somatic cells in the human are, and most other animals are diploid. So everything is diploid. That's in all of the body cells. But in the gametes are haploid. Those are your reproductive cells. Sperm cells and ova are gametes. Ova are your egg from your ovary. And they differ from somatic cells in their chromosome number. Gametes only have 1 set of chromosomes. Human gametes contain a single set of 22 autosomes and then 1 pair of the sex chromosomes, either an X or a Y. So who determines the gender of a baby? Well, girls can only give an X chromosome, but the male can give an X or a Y based on the sperm. So it's really the male sperm that's going to determine whether a baby is a male or a female, right? Which makes you ponder about all those kings who killed all their queens because they wouldn't produce a male heir. So very interesting. All right, meiosis reduces chromosome numbers from diploid to haploid. Meiosis is the one-way process, and it cannot be said to engage in a cell cycle as mitosis does. Meiosis is a sexual reproduction, significantly contributing to genetic variation. All right, so in the end of meiosis, you end up with 4 daughter cells. And you go through meiosis 1, and then you go through meiosis 2. All right, here is a normal human karyotype. I was showing you pictures of this earlier. So we have 23 pairs. So this one is, yeah, there you go. There's your 23rd pair right here. So that's a male, having 1 tall one and 1 short one is a male. That's the 23rd chromosome. Okay, crossing over only happens in prophase 1. And as I said earlier, this gives us a chance for a recombination so that the offspring has a different trait. So you can see what the parental ones look like, and then you can see what the combination is here in the offspring. So you're getting a chance for variation. Mendel has 2 laws. There's a law of segregation, which states that while a parent may have 2 alleles for a certain gene, the alleles are separated from each other during meiosis. So I have an allele from my mom and an allele from my dad, but in meiosis, they're going to be separated apart. So that was when we had the 2 sister chromatids for one and 2 from the other, and they get pulled apart. So your child is not inheriting both of these. The law of independent assortment, unlinked or distinctly linked segregation genes pairs assort independently in meiosis. The way an allele pair is segregated during meiosis has no effect on how the others pairs are separated. So when those homologous chromosomes come up, which is it, this way or this way? And even in the sister chromatids, is it one goes one way and the other way, or is it the other way? It's all independent. So who gets pulled apart and where, and what's the point? It's to lead towards variation. It gives you another chance for random genes. Okay. So you can go back and read through that. All right, here's in our parent generation, we're going to call it the F1 generation. It's called the first felial generation. In the F1 generation, we see a trait, we see meiosis is happening, and we're producing our offspring from this. And that's what we call the law of segregation and independent assortment. So you can see those separating. And you see anaphase 1 and metaphase 2. And we're down here with our 1, 2, 3, 4, or 1, 2, 3, 4 different genes. You can see what the probability of inheriting it is. Next week, we'll be doing some genetics, so I'm excited with that. Independent assorted genes tend to have a 1 to 1 to 1 to 1 ratio or 50% recombination frequency. Independent assortment from meiosis. The 1A produces a recombination of frequency of 50%. The simplest interpretation of such a result is that the 2 genes are on separate chromosome pairs. However, genes that are far apart on the same chromosome pair can act virtually independent and produce the same results. Okay, crossing over produces recombination frequencies lower than 50%. The gene close together on the same chromosome pair, the physical linkage of the parental allele combination makes independent assortment impossible. The genes closer together, the more likely the gene is to be passed together, to be linked. So as we're looking here, a gene here on the top part versus a gene on the bottom. So if these two genes were next to each other, they have a better chance of being stuck together and being able to pass on. If it's something on the bottom, it could have had crossing over that happens and then separated independently. A recombination frequency significantly less than 50% shows that the genes are linked. A recombination frequency of 50% greater means that the genes are unlinked and separate chromosomes. Okay. All right, so when we get into genetics, that's what this is going into, okay? Sorry, right, let me go back here. Okay. We get into genetics. When we do two crosses, we want to know if those genes are linked together or they're not. So what we do is we test cross against something that's homozygous recessive, so we know what all of their genes are. And then we cross with something that's unknown, that has at least one of the dominant traits, we just don't know which ones. And we're able to see on the offsprings who inherited what. And that also lets us see then what was linked together. Okay, so the stunted is the wings, having those little wings right there. Oh, sorry, here. These are stunted wings. Okay, so when we look at data like this, we're able to say, you know, has crossing over occurred? What is the recombination frequency? And are genes linked? Okay. All right, and again, we'll be into this in genetics too. Okay. Okay. So here we have the chromosomes, and they're talking about what traits are in individual places. So this is the short little antennas, or the long ones. This is the black body. This is the gray body. Okay. The shape placement of the eyes, the wings, and the having brown eyes or red eyes. Okay. All right, compare these two processes and how they're similar and how they're different, and which cells are haploid and which cells are diploid. All right, so here we have our parent cell. It doubles in chromosomes. Oh, what is this happening? This looks like crossing over. Okay. They're pulled apart. Who are they? They are the homologous chromosomes. They get pulled apart. And then the sister chromatids are pulled apart. Okay. So and then right here, this is the chromosomes. They've doubled, no crossing over. The sister chromatids line up. All right, so this one we should recognize as haploid. This is meiosis over here. This is diploid, and this is mitosis over here. All right, again, a difference between mitosis and meiosis 1. Meiosis, remember, there is meiosis 1, and it goes right into meiosis 2. And we end up with those 4 daughter cells. Everybody has interphase. So just remember, in interphase, we have the S phase where we double those chromosomes. Remember, in interphase, we have G1, S, and G2. So we're preparing for mitosis or preparing for meiosis. The S phase is the most important because we're doubling our chromosomes here that we need to be able to send to meiosis. All right, meiosis 1, the cell division segregates the 2 chromosomes of each homologous pair and reduces the chromosomes by half. This is longer than most complex processes than prophase of mitosis. because we have crossing over that's occurring in prophase 1. Prophase 1 typically occupies more than 90% of the time required for meiosis. All right, there's the crossing over that's happening. We're to get recombination. So again, we did the drawings that kind of went through all of these and kind of helps you like remember what's happening and what they look like. I hope my drawings help you out with that. Go back through here and read through those notes and definitely watch that video. Okay, exit here. And that will take you through today's and what we're going through our notes today. All right, and let me know if you have any questions. Thanks.